Assalamualaikum dear students, I hope you all are good. Today we are starting with our next topic, that is the mechanism of enzyme action. I'll be explaining like how enzymes function, how do they play their role in a particular reaction. So here is an enzyme. So let me tell you, every enzyme has certain sites at which the substrate binds. These are also called as, earlier these were called as binding sites, and now these are called as active sites. So every enzyme has particular sites for a substrate to bind. Here is the substrate, and the substrate has the conformation, like it can fit into the active site of this enzyme. So every substrate has a conformation that can fit into the active site, that gets attached at the active site of an enzyme. So I already told you that the enzymes are very specific, which means that they act upon a specific substrate. They act upon a specific substrate. So what happens when this substrate upon which the enzyme acts plus enzyme, they combine? What happens, and a complex is formed which is termed as enzyme substrate complex, fine, and later this complex dissociates, and the substrate gets changed into the products. So I'll be explaining this. So here you see, this is the sucrose here and plus water. If you combine the sucrose and water, and the enzyme in this process is invertase, fine, and the enzyme in this process is invertase, sucrose gets changed into glucose, and as you see here, the sucrose gets changed into glucose and fructose. Fine, this is the enzyme invertase. So here is how sucrose plus water bind here at the active site of this enzyme, and later when it's released from the enzyme, glucose and fructose is formed, and this enzyme is invertase. The enzyme in this process is invertase. So let me tell you, mechanism of enzyme action we are studying. Like what happens, the enzyme binds to the substrate, the compound to be catalyzed, and where the substrate binds, it binds at the active site, as I have already told you. So when it binds by means of weak non-covalent interactions, and it does not form particular covalents. In some cases it forms a covalent bond also, but usually the interactions between the enzyme and the substrate are non-covalent interactions, weak non-covalent interactions, like hydrophobic interaction is there, hydrogen bond is there, ionic interaction is there, or sometimes reversible covalent bond. Reversible covalent bond here is like if a covalent bond is formed between the active site of an enzyme and a substrate, later that bond is broken, fine, and the reaction is reversible. So now what happens, the free energy released in the formation of these weak interactions, when a bond is formed, energy is liberated. So when these interactions are formed between the substrate and the enzyme, a large number of you can say free energy is liberated in the process, and that energy is termed as binding energy. And what does that binding energy do? That binding energy is used to lower down the activation energy. In short, you can say when the substrate and the enzyme they combine, fine, the molecules of the substrate come closer. As a result, they can form the bond between them, and as a result get changed into the product. So here, when enzyme plus substrate combine, they form enzyme substrate complex ES, and later on enzyme and product complex is formed, and later what happens, enzyme gets dissociated and product is formed. And let me tell you, enzyme is never consumed in a reaction. Enzyme gets liberated at the last of the reaction, and it's never consumed in any reaction, neither changes its form. So moving forward, let me tell you, there are models of enzyme action mechanism. The first model is lock and key model, or lock and key hypothesis, and this model was given by Emil Fischer in 1894. In this analogy, if you say enzyme is considered as a lock and the substrate is considered as a key. So let me show you how. Here is the key substrate, it's just an example, and here is a lock, it's an enzyme. So it fits it into it. So this is the lock and key model. Like here you see an enzyme, this is the active site of the enzyme, and the conformation of the substrate is such that it forms a key into this active site of an enzyme. They fit like the lock and the key. So here is the enzyme substrate complex. So let me tell you, according to this model, the structure or conformation of the enzyme is rigid. अब इसका जो drawback है, there is a drawback of this model also. It says that the structure or conformation of the enzyme is rigid. ये rigid है, ये change नहीं हो सकता है, and substrate fits into the active site just as the key fits into a proper lock or hand into the gloves. Like the key fits into a proper lock, like we put hand into gloves. So it's that type of model. It suggests the enzyme and the substrate interaction is like that. And this model does not give any scope for the flexible nature of enzyme. It does not say that the enzymes are flexible. It says that the enzyme is rigid, and the active site is pre-shaped. एक्टिव साइट जो होती है पहले से ही उसकी शेप होती है, इट्स प्री शेप्ड. सो दिस एंजाइम this model fails to explain many facts like enzymatic reactions, and most important are allosteric modulators. I'll be explaining later when we study inhibition in tomorrow's topic, inshallah. So let me tell you, so this model has a drawback that it states that enzyme is rigid and the active site is pre-shaped. Fine, it does not give any scope for the flexible nature of the enzymes. Here you see, this is the enzyme, like it is a lock, it behaves like a lock, and here is the substrate. It fits into the like the key. Substrate fits into the lock and forms the enzyme substrate complex. Later what happens? The substrate changes into the product and enzyme gets dissociated from this. So this is the lock and key mechanism. Now moving forward, there is another. You can see that the induced fit or Koshland's model. Koshland proposed this model in 1958, and it is the most acceptable model. It gives scope about the flexibility of the enzyme. Here you can see, here is a this is the enzyme, this is the substrate. Fine. The enzyme modifies its active site, and later when the product is formed, it changes back to its original state. Here it shows the active site of the enzyme shows flexibility. Fine. It shows the flexibility. You can see once again, this is the substrate and this is the active site of an enzyme. When the substrate comes here, fine, it changes its shape, and later when the substrate is released as a product, it comes back to its original shape. So it states, this model states that the active site of the enzyme or the enzyme is flexible in nature. Fine. And let me tell you here, this active site of an enzyme, it contains certain amino acid residues. What is their job? They align themselves according to the substrate. Here you can see, this is the active site of an enzyme. Here these amino acid residues, they align themselves according to the substrate. So it gives flexible nature to the active site or in whole to the enzyme. So Koshland's model is much accepted model as compared to the lock and key model. Lock and key model fails that it explains that the enzyme is rigid and the active site is pre-shaped. But here the model fails. Now what Koshland's model states? It states that the active site of an enzyme is flexible and it changes its nature according to the substrate. It changes its conformation according to the substrate and later releases the product. Fine. So this is all for the today, and I hope it's clear to you. If there is anything, please let me know. Stay blessed and stay safe.